Welcome to the Scientific Method webcast. As always, I'm going to fly through this PowerPoint fairly quickly. Feel free to stop and go back as needed to complete your notes to understand these learning objectives. So, scientific method, what is it? If you were to look this up in Google, you'll come up with I don't know how many different responses, but there'll be a variety of models out there. Today we're going to use 5 basic steps to the scientific method. So real quickly, ask yourself, what is science? What is science? Coming up with an idea for science, you may have listed a couple of science classes, and those are just different disciplines within the process of science. Science truly is an organized way of using evidence to learn about the natural world, and that's important. So when we're studying biology, we're using things that we've observed and tested to help us explain whether it's the human body or ecology. We're using science to explain that. So things in the past, things in the future we could discover. We're using this process of learning and discovering. It's called science. So the first step of the scientific method is our observation. So we make an observation, which is simply using our senses to gather information. You may think of this as sight a lot of times, but you can use smell, your sense of touch, your sense of taste. Try not to use your sense of taste too much in science. This leads to a question, because you're wondering, what's happening here? I'm seeing something that's interesting to me. What's going on? As we talk about this, there's some different types of data or observations we can make. One is quantitative. If you look at the word quantitative, it kind of sounds like quantities, and that's exactly what it is. It's a measurement of numbers. So some examples of quantitative data would be temperature, degrees Celsius. In science we want to use the metric system, so Celsius. Maybe we want to do volume, length, mass. Right? Those are all things that we can measure in numbers. Quantitative things do not have numbers attached to them. These are characteristics. So maybe color, texture, etc. These are other descriptive words we could use. In science, we really like to use quantitative data to use as supporting evidence. It's easier to use numbers to tell us more about a phenomenon we're trying to understand. Second step, hypothesis. You may have heard a hypothesis as an educated guess. It is kind of an educated guess, but that's an elementary term. We're going to bump it up a notch here for high school. Possible explanation for an observation based on prior knowledge and or research. So a true hypothesis, a lot goes into it. True scientific hypotheses, there's a lot of background work before a hypothesis is ever made. Try to write these in an if-then statement. So, should a hypothesis ever end in a question? I don't think so. It needs to end in a period. It's a statement and it's testable. Very important. Okay? So we'll maybe practice on these in the future. But here's a quick example. If the amount of sleep is related to GPA, then getting more sleep will result in a higher GPA. We can do a quick survey of 500 students and ask, how much do you sleep? And what's your GPA? We could crunch some numbers and figure out if there's a correlation between sleep and GPA. Step number 3 is a controlled experiment. It's called controlled because we're only testing one variable at a time. So we're controlling everything, including the variable that we're testing. The word for that variable is independent. So this is the one thing we want to test. Sometimes it's called the manipulated variable. Because we're actually changing it. We want to change it. And the response to that manipulation is our dependent variable. So response to the independent variable. It's important that you define both of these in your scientific experiments, and we're going to do a little practice identifying those here on the next slide. So I have 3 examples here. What I want you to do is to create quick number 1, 2, 3, write down IV for independent variable, DV for dependent variable for all 1 through 3. And I want you to think about this sentence. The blank, we can fill this in with dependent variable, depends on the blank, and that's the independent variable. If you have these correct, this sentence will make sense. If you flip-flop these, then the sentence will not make sense. So use this as you practice on those 3 examples. So go ahead now, press pause, and practice. See if you can identify the independent and dependent variables based off of your definitions. Okay, you should have had plenty of time to practice. Now let's see what they are. I'm going to use a couple things here. Independent variable, I will use a circle, and a dependent variable, I will use an underline. First one, an experiment was set up to see how the amount of fertilizers affect the number of tomatoes. Our independent variable is the amount of fertilizer. We can change that. We can see, put so many milliliters of fertilizer into various plants. Our next thing here, we want to measure something, so our dependent variable is the number of tomatoes harvested. This is actually a something we can measure. So the number of tomatoes depends on the amount of fertilizer. Number 2, a field study examines the amount of moss growing on north or south facing rocks. So our independent variable is which way the rocks are facing. Okay? Which way the rocks are facing. And our dependent variable is the amount of moss. If you fill in the sentence, the amount of moss depends on which way the rock is facing. If we were to flip that, the way the rocks are facing depends on the amount of moss. That doesn't really make sense. The moss isn't going to make the rock change which way it's facing. Third one here, a set of observations records how much weight dog gains in the first year. This one's a little tricky. Our independent variable is first year. We could possibly measure this monthly. And our dependent variable would be the weight of the dog. So maybe we're looking at how many kilograms it gains in the first year of its life. A couple other things we need to consider here are control group and a standardized variable. So our control group is something that remains constant to see if any changes actually occur. It's important we have this, otherwise we're not sure if our manipulations are different than what's normal. And then our standardized or controlled variables, sometimes also called constants, they are going to be what remains the same in each of our test subjects. So in here, our control group would be no fertilizer, and our standardizing variable, no fertilizer, because we can see how many tomatoes grew without fertilizer, and we can compare that. And if we see there's a whole bunch of tomatoes, maybe there's 10 times the amount of tomatoes when we have a certain amount of fertilizer, then we know some results occurred. Standardized, we want to keep the same, so we want the same soil. We want the same amount of water. We want the same temperature, light, pretty much all the things that plants need to grow. The only thing that we're changing would be the amount of fertilizer. Perfect. Next step, record and analyze data. So this is the part where we're actually probably recording during step 3 as we're doing our testing, and then we need to take a look at our data. But an easy way to do this is a point where we make graphs in tables. Typical bar graph, you guys have all made those before. This compares 2 or more sets of data, and line graphs are going to often be used as change over time. So we want to see, this would be a good one for our tomato plants because we can have fertilizer A, no fertilizer, and we can see if there's a difference. Here we want to look at the dog's weight over time. Maybe we see something like this, and all of our months, January, February, March, April, May, would be down here, and here's the mass of the dog over here. Okay, good way to look at that. There's other graphs and charts out there. Maybe you want to make a pie chart. We'll go into that in our next webcast. Next step, conclusion. State whether the data supports or does not support. It's not proves or disproves. Make sure that we support or does not support. Very important. And also when you write your conclusions, we want to refer to the data. Okay? This stuff right here, this is evidence. Remember, talking about science is a way of using evidence to understand the natural world. Well, here's your evidence. Use that in your conclusions. Finally, word theory. We've all heard the word theory. This is a big time word in science. It's very important. A scientific theory is a well-tested explanation for a natural phenomenon. It's supported by multiple experiments. Can always be disproven if new evidence arises. So we have a couple examples here: theory of evolution, cell theory. If we find something new in the later in science, we can go back and change it. That's what science is all about. Don't use this. It's just a theory. Well, actually, theory is pretty important. So it's never just a theory, especially when we're referring to science. Or have you ever heard this? I have a theory for something, which is maybe just an explanation. Don't use this word because it's not, in true sense, what a theory is. It actually decreases how important the word theory actually is.